So that's how they used to record or a single neuron signal. And what we do is they just put the recording pipette into cell neuron itself, and then once we put this little suction here, and we can kind of rip it out, right? So for this case, this is the inside out recording, and what they do. So now that you have this like an ion channel facing this way, where so you use this ion channel to kind of like get the signal through the pipette and record the signal that way. On the other hand, what you can also do is you can kind of rip apart this cytoplasm, the cell wall. No, not cytoplasm, but the cell membrane. And then if you like take it apart, like they're kind of stuck there, and then like sometimes after they kind of like anneal itself, they like repair their cell wall, and then now you have this like a cell membrane. But what the difference is that the orientation or the direction of this this ion channel, as you can see. So now what you do is you do like outside out the cell recording that way. So now this is for extracellular extrophysiology, and you can put like electrode on different places, not only just you can put in an axon, dendrite, or cell body, etc. And we see a different type of signal where depends on the location we put. So for example, these two are like extracellular, whereas this top one is intracellular, and we see the difference in electrode or the signal itself. And go in more into the extracellular electrophysiology, for in vitro type, we can do this in micro electrode array and put neurons on top of it to read the signal that way. Or we can do more of an in vivo approach, where the electrode itself is like on top of the brain of mouse, and then we can record the signal that way as well. The problem of this electrophysiology is that it's really hard to interpret the signal, and like so in order to like sort the spike, we have to classify with either like low pass filter, histogram, Gaussian filter, etc. You have to just like, it's just really hard to classify what type of like spike it is. So there was the different type of filters that I just mentioned. So we have this raster plot over here, and then now we can do like different histogram with different beam sizes, so different resolution, I guess. And then we can either do like a low pass filter like over here, or we can do like Gaussian filter to kind of smooth out this signal. So this is a visual pathway that goes from our eyes to, and the signal kind of crosses over here in the middle, and they eventually go to this lateral geniculate nucleus, which we'll call LGN. So we see that the optic tract, and it follows to LG, neuroglycinal body, LGN, and then optic radiation, and it eventually goes to primary visual cortex. This is the retina, like the back of the eyes, really. And the one thing kind of interesting is that this is where these are the sensor of the light signal, but they're actually at the back of the eye, and it's like covered by their wires, I guess. But the reason being is that they need like a oxygens and stuff, and they're supplied by the like over here in the back. So that's why they're like located at the back apparently. So cone and like rod, they have like a different features. Like cone is better for daylight, and they're specialized in color, and rod is for a low light situation with like a kind of like black and white kind of they see black and white. So photoreceptor membrane voltage recording, so when there's a signal at like time 0, depends on the intensity of the light, whether it's dark or bright, they show a different membrane potential or peak voltage. However, one thing to notice that they're this time scale from 600 milliseconds is quite long for them to go back to their steady state. So it's kind of like slow process. Something I mentioned before, rod is like, so the rod and cones, they're a little different. The cones, which is specialized in like a daylight and color, they're fast and they have a characteristic of bandpass filter, whereas rod is the other way, slow and has a low pass filter characteristic. And so assuming that we have this kind of light stimulation where there's a light just in the middle, it's brightened, and this is like a time scale, time axis, and then this is like a voltage. So for receptor, they respond to it, and then so the membrane voltage goes up, and horizontal cells, bipolar cells, they show like different characteristic. Amacrine cells, it seems like they kind of note the beginning and the like end of the light signals. That's when they like fire. Ganglion cells, so there's a on center and off center. So for on center, they like fire during the time when there's a light stimulation. So this is like in the middle is when the light is present, a time that when the stimulation is given basically. And the off center is depressed when there's a light signal. And this is the right column is a different style, where there's like a donut. So in the middle there's no light, but in the kind of circular shape, like a kind of like an influencer light type of thing, they have a light. And we see that it's like the opposite now. The off-center one has more, they're firing, whereas the on-center isn't firing. So now we have this on-center. This is on area and this is off area. We can kind of see. So this line is indicating the time that the light stimulation was given. So in this field, let's start with the simplest one. So the surround illumination, there was this on spot in this smaller circle. It was kind of like rather darker than the outside exterior where the off area. So we see that when this light was given, there's a depression where there's no signal, and then afterward, it is kind of there's more signals. And then for diffuse illumination, so where everything is kind of bright, it shows pretty good signals over there. And for the central illumination, so now this off area is like off, and then only the on area is like really bright. And we see they have higher activity. And for peripheral spot, doesn't show here. Central spot, it is like a little bit on, but not all of them. So there's a little bit of activity going on, but not as intense as like the other one, like this one. So this is like off center. So this is like the flip. We have now flipped it. So for there's a very bottom where everything is bright. The off area is now like on, I guess. Oh well, let's go with this one. So this off area is like on, right? Like so it works the other way. So now this should be repressed because it should be activated when it's like dark on the center. So we see that there's depressed when the stimulation is given, and this is kind of like the opposite of it. When the off area is like off, so it should be activated, and it is activated indeed. And so on and so forth. So this is kind of like confusing, but it is what it is. So this is kind of like the very simple wiring diagram of the like retina. So when there's a receptors and they go to horizontal cells, but some of them they go direct path to the bipolar cells, and bipolar cells are kind of like a activator, whereas for the indirect path they kind of work as an inhibitor. So direct excitatory component is D over here, this one, and the indirect inhibitory, this like gray one, is I. So when we like add them up, we kind of have this kind of voltage going on, and we call this apparently like dog function, which stands for difference of Gaussian functions. So when we add them up, we see the difference, I guess. So this is how they actually get the data from it. We sedate the cat, and then we keep their eyes open. We put electrode on their brain and let them see stuff, and we record the data. And we have this like a light through a projector or flashlight of some sort. And notice that this is like it's like a light, it's like a rod shape, right? It's like a stick. And there's a reason behind this. We're gonna talk. So the orientation really matters for this one. Apparently, they found it by a mistake. There's a higher, much higher activity at the certain orientation of the light, and it's really interesting. For example, like in this example, when the light is given in this the vertical rather than in horizontal, there's a lot higher spike rate, a lot higher neuron activity. And what it's suggesting is that, so this is like the in and out, on and off center that we were talking about. What it could imply is that in the retina there's a receptive field is oriented that way, where they're all kind of connected to the LGN, and then goes to one cell in the cortex. So if the light were to be presented this way, and it's hitting all this retina and activating, so all of these LGNs are activated and sending strong signals, and we have hence we get a stronger signal reading from electrode. However, let's say the light is presented on the other side, other orientation. Now it's maybe hitting at max 1, maybe 2. So compared to all 4 active signal, which was this direction or orientation, if we're getting the other orientation, well, we're only activating 2 signals, right? So the signals are a lot weaker, and hence we get less strong signal reading from the cortex or the electrode. But that's the hypothesis, and it's not proven apparently. This is a 3D representation of it's not in real life. We're the on and off center, where it was depicted as a circle, but in reality it's kind of elongated. So that's how it is. So this is activating, and this is depressing characteristic on different cortex. And the wiring